Real honor to be here. It's a great pleasure to talk to you this morning. I'm going to be talking to you about the Healthy Plants, Healthy People, Healthy Planet project, which is a joint project between the John Innes Centre and the Sainsbury Laboratory, which are on the Norwich Research Park in the United Kingdom. So let me tell you first a little bit about the John Innes Centre, and then I will tell you about the Sainsbury Laboratory, of which I'm privileged to be the director, and then I'll go on to talk about our vision for the future and the Healthy Plants, Healthy People, Healthy Planet project. So the John Innes Centre is a long-established, world-leading centre in plant sciences. It has a century-old tradition in genetics. In fact, the term genetics was first coined by the first director of the John Innes Centre, William Bateson, in 1904. He was writing a letter and he was thinking of a new term for heredity, and he coined the term genetics. And really, genetics has underpinned so much of what the John Innes has been about. And by genetics, I mean in its widest sense, the means by which crops have been improved over many years by plant breeders, the underlying genetics of that. Many of those discoveries were first made at the John Innes, and that really underpins much of the work that still happens across the John Innes Centre. And there's been huge advances in genome sequencing, which now mean there are unprecedented opportunities for crop improvement throughout the next century. And in fact, harnessing that technology, harnessing the genomic technologies will enable us to really make a step change in agricultural productivity, but also to do so in a way that ensures there are much lower inputs into agriculture, because we need to make agriculture much more sustainable in the future if we're to feed the world's growing population, but also to do that in a way that is truly sustainable. And our aspiration, of course, is to get to a point where there is fossil fuel free farming, the ability to grow crops without the use of fossil fuels. The John Innes Centre also is involved in genetics and environmental interactions, and understanding their importance in conditioning plant and microbial development. And these are complex issues requiring an understanding of the complicated ecosystems that exist within soil and within field conditions under which crops are grown. And that in future will require an enormous new venture in trying to understand that using technology such as artificial intelligence. So the John Innes has many groups associated with both plant and microbial sciences, and has been responsible for many world-leading discoveries over the last 100 years. These include our ability, or new varieties in terms of wheat, barley, potatoes, many new varieties which have emerged through science of the John Innes Centre. But also on the microbial side, new antibiotics have been discovered, new high-value products that are used, that are produced by microbes, have been produced by the pioneering genetics at the John Innes Centre. Now, the Sainsbury Laboratory was established in 1987, and it was established with a focus on plant microbe interactions. It was designed to take advantage of those huge advances that have taken place in plant genetics and microbial genetics at the John Innes in the preceding decades. And the Sainsbury family charities took the decision because they believed that the time was ripe to make fundamental new discoveries into the biology of plant microbe interactions. And what the Sainsbury Laboratory is really focused on has been the study of plant immunity. In fact, at the time the Sainsbury Laboratory was established, we really didn't know very much about the plant immune system. We didn't really know whether plants had anything akin to an immune system in the same way as humans, for example. What we've learned is, in fact, that plants have immune receptors. They're able to perceive microbes, and they're able to then defend themselves. And those immune receptors are the products of the resistance genes that plant breeders breed into our crops. So an understanding of those means that we're better able to deploy existing resistant specificities, things that we know of from plant breeding, but also importantly to discover completely new means of protecting plants. And that's been the focus of the work at the Sainsbury Laboratory over the last 30 years or so. It's a small focused research institute. It's made many fundamental discoveries in plant immunity. The first immune receptors were discovered at the Sainsbury Laboratory, but also it's discovered technologies such as RNA interference, a mechanism which is now used in many important human medicines. In fact, a whole class of medicines are involved, which are involved in gene silencing, which were discovered based on the discoveries first made in plants by scientists at the Sainsbury Laboratory, including most notably Sir David Balcombe. The Sainsbury Laboratory is now applying its research to enable plant diseases to be controlled using plant genetics so that we can reduce chemical inputs. That involves a whole raft of different genetic technologies, including marker-assisted breeding technologies, genetic modification, genome editing, a whole variety of different approaches that can be taken to deploy resistance in the field, and to do that in a sustainable way that leads to durable resistance. Let me tell you now about the Healthy Plants, Healthy People, Healthy Planet program. This is a vision that we have for enabling our science at the John Innes Centre and the Sainsbury Laboratory to truly address the global challenges that are facing us, particularly in the area of food and agriculture, the whole subject area of the work that we're doing today, that we're showcasing today at in this New Scientist event. What we really would feel that there's a great potential to do is to, through our understanding of plant and microbial sciences, through our understanding of the way that plants interact with each other, the way that which plants interact with microbes, that we'll be able to use that understanding for the good of humanity. We'll do that because by having healthy plants, we can have a rich and nutritiously rich source of food for the planet. And we can also develop technologies which will enable much more sustainable agricultural development. Work at the Sainsbury Laboratory associated with plant immunity can lead to plants which are durably resistant to a whole host of different diseases. And work at the John Innes Centre will look at resource allocation, trait development, and all of the constituents of yield that are so important, and our understanding of both inputs to crops in terms of fertilizers and growth conditioning agents, but also their ability to be able to yield highly with much reduced inputs compared to contemporary agriculture. So in order to do that, we need to have completely new ways of working. We need to work together in a totally different way. We're going to need to be able to carry out our science in a way that isn't siloed into traditional disciplines, but instead is truly interdisciplinary. So we need biochemists, cell biologists, microbiologists, computer scientists, all working together in a seamless way, in a way in which that they're tackling the problem using a whole diverse set of different experimental and computational approaches. And that's the vision that we have within HP3, to enable us to actually harness all of that technology in a completely new state-of-the-art facility that will enable us to work together in a much more integrated way than is currently possible, and indeed currently possible in most laboratories as they're currently designed. So it's a radically new departure for both the Sainsbury Laboratory and the John Innes Centre. We want to work together in a much more integrated manner. This will involve the use of platform technologies in bioimaging, in advanced biochemistry, and in computational analysis, coupled with advanced areas such as bioimaging. So by bringing all of these technologies together, we feel that we can make a step change in our ability to address these fundamental processes around plant sciences and plant and microbial sciences in future. So that's the vision that we're setting out. And I've been joined by my colleagues now on stage, and we'll be very happy to answer any of your questions around the HP3 project, about our vision for the future, and also about the current science in both of our institutes. So thank you very much.
Could I just add to what Nick has said, if that's okay? Thanks, Nick. And I think it shows very much how the John Innes Centre and TSL are really joined at the hip, because we are you're well able to speak with for the John Innes Centre and some of the notes that I want to make. But it's really an exciting time. Just to re-emphasise what you've said. I mean, I think, you know, a lot of people listening will know about the Human Genome Project about 20 years ago, the Human Genome. We had a reference genome for Arabidopsis, a model plant. We're now getting into this amazing era of diversity where we can exploit or develop our understanding of what makes individuals within a species different, and that has enormous implications, I think, for the development of crops that are both resilient to climate change and that provide minimal impact on climate and as you were saying earlier, require fewer inputs and resources, and which enable agriculture to harmonise with biodiversity, which to tell the truth, it hasn't always done. But I think we're entering into this exciting new era.
Thanks for covering while we joined in, Nick. I don't know how much you talked at the beginning about the kind of topics that we're going to be covering all day here at New Scientist Live, and how they're really well matched to the kind of healthy plants, healthy people, healthy planet vision. But we'll be in our room, the John Innes Centre Healthy Plants, Healthy People, Healthy Planet room all day. So we know that some of you are having problems with your audio. I'll write in the notes this same as well, because people might not be able to hear us. So if you do have any questions or you want to talk to anyone from both the Sainsbury Laboratory and the John Innes Centre all day, we'll be in our room. So yeah, please join us and we can talk more about the vision and about the science and technology that we're working on here in Norwich. We have got a video to play you, but I don't know if that might be a step too far with the technology, but I could give it a go now. I'll see if I can play it now. So I'm going to try and I hope the audio comes through. Oh, because I'm on my laptop. Sorry, everyone. Maybe we'll cancel this. We'll be playing the video.
Should we take questions, Felicity?
Yeah,
I mean,
that's a good idea.
And leave the video up there with a link. But there's some really interesting questions coming in, and I'd be very keen to answer them, as I'm sure Nick would.
Yeah.
Do.
So I've just seen one come up really recently. I guess this could be probably to Nick, but also to Dale as well. So what will be the impact of HP3 on of these new plants on subsistence farmers?
Well, we work a lot with smallholder farmers through much of our work in low and middle income countries, and it's important to say that both of our institutes are really exist for the public good. Of course, we work with a range of organisations, both in the private sector and the public sector, but a lot of the work that we're doing is actually funded by charitable organisations, and we're trying there to work on, for example, issues. I can give you a direct example. We're working with farmers in sub-Saharan Africa who are dealing with the rice blast outbreak, and what we're doing there is using marker-assisted breeding to try and introgress resistance genes into locally adapted cultivars which are used by smallholder farmers, and those will then be distributed free to those subsistence farmers. And that's I think a typical example of the ethos that exists at both the John Innes and the Sainsbury Laboratory. We want to work with smallholder farmers as well. We want solutions that are available for them as well as the big commercial operated farms in the developed world.
Could I just add to Nick's comments, which I fully subscribe to? We realise, I think, both the Sainsbury Laboratory and the John Innes Centre realise that as scientists, we can't address issues with subsistence farming on our own. We need the involvement of social scientists. So we are in the process of launching a Norwich Institute for Sustainable Development in collaboration with social scientists at the University of East Anglia. And this will be a really joined up project which involves detailed understanding of the biology of agriculture, but also detailed understanding of what social issues are and how the sort of science that we do can be deployed ethically. And that's a key word in terms of how we want to move forward.
Great, thanks both of you. We've got quite a few questions coming in around what John and his compost.
Yes.
I can probably answer that one. So the John and his compost, we don't actually make it. We developed the recipes. So there's 4 different recipes which were developed between 1932 to 1937 and were released during the war as part of the Dig for Victory war effort to the public. And actually the recipes were taught in schools until the 1950s. You were taught how to make compost in school. We gave it away for free. We've never made any money from the compost as an institute. And essentially manufacturers make compost from our recipes, and some of them do still contain compost. I know that the industry have been working really hard to reduce peat use in compost, and I think there is now a goal to eliminate it from commercial compost in the next, I think it's 5 to 10 years. I'm not entirely sure on the dates, but you can go to the Compost Association website and find out more about that. But the story behind the compost here at John Innes is quite interesting because it was a result of a huge scientific failure. One year they were growing 7,000 different lines of primrose to breed new colours and types of primroses for gardeners, and essentially all of them failed one year because of poor soil and poor compost. And this led to a research programme through the 1930s that gives us the compost we see today. And they invented things like sterilisation of the soil, so they heated the peat and the soil and the sand that they used to mix together to eliminate any bacteria or microbes in the soil, which gives you a much better germination rate and growth rate. And then they also worked out the components that you need, so the nitrogen, the phosphorus, and the other minerals that you need in the soil. So yeah, we are very keen to reduce peat in compost, but it's not really our responsibility, I'm afraid. So that's a short story. There's loads more about the compost on our website. If you do want to find out more about the history of it, just search on the website for compost. I didn't know if you, Nick and Dale, wanted to add anything about compost.
No, I'm keen to.
Felicity, there's loads of really interesting questions coming in, and I'm wondering whether we can just put those on we're not going to be able to answer those in the remaining minutes. There's some really stimulating things. I saw some interesting things come in.
So if you, Dale, if you want to choose one you've just seen and answer that now, I'll take the ones we've got coming in and we'll answer them in the room throughout the day. Okay. So did you see one you particularly thought was interesting that you wanted to answer?
There's a couple of come up on GM. And in fact, there was a very interesting one about being generally positive, but what are our approaches to using terminators and markers and so on. And I'm a big fan of thinking that, GM, of course, like all agricultural technologies, needs to be regulated. It needs to be risk assessed, properly risk assessed, but risk assessed by what you've done and what you produce in terms of a trait, not in terms of the mechanism by which you got there. So if there is the possibility of horizontal gene transfer, which is deleterious with what you've produced, then that risk has to be assessed. If there is the possibility, for example, of using genes in order to of using genetic technologies to generate disease resistance, which reduces the use of agrochemicals, then of course we should take that really seriously but not disregarding any potential risk. That it has to be regulated in a sensible way.
I think there's also some interesting questions about genetic diversity, and I think that's something that's really important to all of the research being carried out at both institutes. What we're finding, of course, with genome technologies is that there's actually a vast array of genetic diversity available, and that can be deployed very often through conventional breeding or through genome editing approaches, and that's a hugely valuable resource. So I think that the questions which are around the importance of genetic diversity in resilience, in terms of our ability to grow crops better, we absolutely agree with you. That's exactly the approach that we want to take.
Great. One of the questions we can answer quite quickly, I think, is what crops will you be working on? Currently, wheat and rice would provide 37% of the world's calories. Can this be good?
Well, it probably isn't good, but it's still a fact that there's about, I think, 6 species provide almost 60% of the calories on the planet. So of course we work with those important crops. We work a lot with wheat, with rice, and with a variety of other cereals. But importantly, we work a lot with other crops too. We work a lot with vegetables. We work a lot with crops which are grown in low-income countries. And I think the point again is about genetic diversity. It's trying to look at the diversity that exists and to try and understand how those crops evolved to actually think about their genetic potential. So in order to make us more resilient and our food systems to become more resilient.
Yeah, I think one of the really interesting sort of cross-park collaborations that's happening is with Tropic Bioscience, who are working to improve banana genetic diversity and their resistance to banana wilt, and that's in collaboration with both scientists at the TSL and the John Innes Centre, and that's essentially a crop that's completely dependent on one genetically similar variation that we don't really draw upon the genetic diversity that's there in bananas. We just eat the one type, and that's a really good way for us here in Norwich. We can bring in companies like Tropic Bioscience. They co-located here to work with us and our genetic understanding and the work we've done with looking at the diversity that is out there, and they've been really successful and hopefully going to be able to help secure banana crops in the future. There's another question. It's a bit confusing because there's questions in the chat and also questions in the Q&A. So if you can put your questions in the Q&A section, which is on the right-hand side of the screen, we can see them a bit easier. And I think we've got 1.5 minutes. So if anyone, Nick or Dale, if you've got any burning questions you want to answer, please jump in.
I did just want to follow up on GM. There's a really interesting one come in on that. It's in the hands of commercial organisations. That has been the case, and that was the big debate. And hopefully, we're around the turn of the century. Hopefully, people will be listening to Mark Liner speak later. But it's not universally the case, and it doesn't have to be the case that our food supply is in the hands of large commercialized organizations. That's a societal issue. We can, as scientists, we can only put potential solutions on the table, and that's why we need society to be involved in terms of deciding what they want to adopt.
I think there's a very popular question from Tom McNamee, which is about why breeding resilience in plants is better than pesticides. I think that we just have to take a very measured look at this. Of course, pesticides have been important in our ability to grow lots of food around the world. If there are going to be chemicals used in farming, we need to make sure that they are as safe as possible, that they have the least number of residues, that they can be degraded in the environment safely, and they don't have off-target effects. The science that we're doing, of course, will help our understanding of those processes as well. But I think we have to look at this in terms of the long-term sustainability of the planet. We can't allow agriculture to remain so fossil fuel dependent. We have to lower inputs into crops for reasons of sustainability. And that's why we're interested in trying to find what are essentially natural approaches to this through understanding the genetics of crops and the diversity that exists in their ability to resist insects, to resist fungi and other pathogens, and indeed other abiotic traits. So we take a measured view on that. So I think that's a really important question, but we have to be realistic about the climate emergency and our responsibility to make agriculture more sustainable.
Yeah, absolutely. I think that's really important. So we've run out of time now, I'm afraid. So there's lots of other talks starting now across all the stages, and you can join us in the John Innes Centre room. I don't know if Nick and Dale have got time to join us there, but there'll be lots of other scientists joining throughout the day. And one of the questions that's just come in is about what are we doing to bring young people into this kind of industry and this kind of area? And we've actually got lots of our young scientists joining us in the room today. So we've got about 10 young scientists joining us, over from PhD students up to postdocs, and then we've also got some of our more senior scientists as well. So come and ask them your questions in the Healthy Plants, Healthy People, Healthy Planet room. Sorry about the technical difficulties at the beginning, but enjoy your day at New Scientist Live. And thanks, Dale and Nick, for giving us some insight into HP3.
Thank you all. Thanks for the questions. Thanks very much.
Thank you, everybody. Really stimulating session.